With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello everyone, the question is with respect to the titration of acid and base, answer the following question. Which solution is taken in the flask? So in this question they are talking about titration, and with respect to the acid-base titration we have to answer the question given here, in which they are asking us which solution is taken in the flask. So first of all we will see what is titration. So titration is a technique which is used to determine the concentration of an unknown solution with the help of a concentration of a known solution. Okay, so technique to know the concentration of an unknown solution with the help of the concentration of the known solution. That is, we have two solutions. One's concentration we know, so with that concentration of the known solution we are going to determine the concentration of the unknown solution whose concentration we don't know. Okay, so in this titration they are talking about acid-base titration, in which we use acid and base, and one of the concentration is known and the other one we don't know. So what we do is, here in acid-base titration neutralization reaction takes place. So with the help of neutralization reaction we determine the concentration of the unknown acid or base, whatever it is. Okay, so for that what we do is, the solution whose concentration is known, we take the solution in the burette. So the known concentration solution is taken in the burette. Okay, and the unknown concentration solution, the solution for which we need to find out the concentration, is taken in the flask. Okay, it is taken in the flask. Now in the flask we add some indicator like phenolphthalein for an example, and then we start the titration by letting the solution in the burette drop into the flask by drop. Usually the solution that is taken in the burette is acid, okay, and the solution that is taken in the flask is base. So then we let the acid fall drop by drop into the base, and when the neutralization reaction has taken place, just after the neutralization reaction the phenolphthalein or the indicator will change color, and so we will read the reading in the burette, and then we will calculate the concentration of the unknown solution here. Okay, so that is how we do titration, and the solution that is taken in the flask is base, or you can say the unknown concentration of the solution. So that was the explanation for the question. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.